Lesson 1. Introduction to Muscle Tissue Types. Welcome. As you settle in for our first session together, I invite you to find a position of absolute comfort. Adjust your shoulders, let your hands rest loosely, and allow the day's tension to simply evaporate. You are entering a space of quiet learning, where the mind can remain curious even as the body begins to drift towards sleep. Tonight, we begin a new journey. We are turning our gaze inward, past the skin, past the framework of the skeleton, to look at the engine of the body, the muscular system. It is a system of movement, of warmth, and of life itself. Before we can understand the mechanics of lifting a weight or the rhythm of a heartbeat, we must first understand the materials we are working with. In the study of anatomy, it is easy to think of muscle as a single thing, the meat of the body, the flesh that moves the bone. But the truth is far more elegant. The human body is actually home to 3 distinct families of muscle tissue. Each has its own unique structure, its own specific location, and its own special purpose. They are known as skeletal muscle, cardiac muscle, and smooth muscle. Tonight, we will take our time to meet each of them, to understand their personalities, and to see how they differ under the microscope and in their function. But before we separate them, we must first appreciate what unites them. What does it mean to be muscle? Regardless of whether it is beating in your heart or blinking your eye, all muscle tissue shares 4 fundamental properties. These are the 4 pillars of muscle physiology. Let us walk through them slowly. The first property is excitability. Imagine a calm lake waiting for a pebble. Muscle tissue is like that lake. It is waiting for a signal. Excitability, also known as responsiveness, is the ability of the muscle cell to receive and respond to a stimulus. Usually, this stimulus is a chemical messenger released by a nerve cell. When the muscle receives this chemical whisper, it transforms it into an electrical signal that travels along its own surface. It is a listening tissue, always attentive, always ready to hear the command to act. The second property is contractility. This is the feature that sets muscle apart from all other tissues in the body. Bones cannot shorten, nerves cannot shorten, but muscle can. When that electrical signal we just mentioned is received, the muscle tissue possesses the unique power to shorten forcibly. It pulls. It generates tension. This is the engine of movement. It is the ability to transform chemical energy into mechanical work, closing the distance between 2 points. The third property is extensibility. This is the ability to be stretched. Think of the muscles in your hamstrings when you bend over to touch your toes. They must lengthen without damaging themselves. Muscle tissue can be stretched beyond its resting length to a certain degree, allowing for a wide range of motion. It is pliable, yielding, and forgiving. And finally, the fourth property is elasticity. If you stretch a rubber band and then let go, what happens? It snaps back to its original shape. Muscle tissue does the same. After being stretched or contracted, it has the ability to recoil and resume its resting length. This elasticity is vital. Without it, our muscles would remain slack and distorted after every movement. Excitability, contractility, extensibility, elasticity. These are the shared traits of the materials we are studying. Now that we have established this common ground, let us branch out and explore the 3 distinct types of muscle tissue, beginning with the one you are most familiar with. Part I. Skeletal muscle. Let us turn our attention to skeletal muscle. When you look in the mirror, or when you visualize the human form, this is what you see. These are the muscles that attach to the bones of your skeleton, sculpting the contours of the body. The biceps of the arm, the quadriceps of the thigh, the pectorals of the chest, these are all skeletal muscles. The defining characteristic of skeletal muscle is that it is voluntary. This means it is under your conscious control. It waits for your command. If you decide to wiggle your toes right now, your brain sends a signal down the spinal cord, out through the nerves, and tells the skeletal muscles in your foot to contract. They obey the will. They are the avatars of your intention in the physical world. Of course, they also work subconsciously, keeping you upright against gravity, for instance, but they are always capable of being overridden by your conscious mind. If we were to place a sample of skeletal muscle under a microscope, we would see something quite striking. The cells do not look like the typical round cells you might imagine. Instead, they are long, cylindrical tubes. Because they are so long, some running the entire length of a muscle, we often refer to them not as cells, but as muscle fibers. The most famous visual feature of skeletal muscle is its pattern. Under high magnification, you would see alternating light and dark bands running across the width of the fibers. These bands are called striations. They give the muscle a striped appearance, like a ladder or a finely woven fabric. These stripes are not just decoration. They are the result of the precise, geometric alignment of the proteins inside the cell, the actin and myosin, which we will explore in later lessons. For now, simply remember, skeletal muscle is striated. It is ordered. It is disciplined. Another unique feature of skeletal muscle cells is their nuclei. Most cells in the human body have a single nucleus, a single control center floating in the middle. But skeletal muscle fibers are giants in the cellular world. During development, many smaller cells fuse together to create these long fibers. As a result, a single skeletal muscle fiber is multinucleated. It contains many nuclei, and because the center of the cell is packed tight with those contractile proteins, these nuclei are pushed to the edges, sitting just beneath the cell membrane like onlookers at a parade. So, when you think of skeletal muscle, remember these three keys. It is striated, it is multinucleated, and it is voluntary. It is the tissue of action, capable of tremendous power and rapid contraction, though it can also tire easily, requiring rest to recharge. Part 2. Cardiac muscle. Now, let us leave the limbs and the torso and travel deep into the center of the chest. Here we find the second type of tissue, cardiac muscle. This tissue is found in only one place in the entire body, the walls of the heart. In the language of anatomy, we call this the myocardium. It is a rare and precious material designed for a singular, relentless purpose: to pump blood continuously from before your birth until the very end of your life. Unlike skeletal muscle, cardiac muscle is involuntary. You do not need to think about your heartbeat. You do not need to wake up every second to command it to contract. It follows a rhythm set by the body's subconscious autonomic nervous system and its own internal pacemaker. It is a faithful servant that requires no supervision. Visually, if we look at cardiac muscle under the microscope, we see that it shares some similarities with skeletal muscle, but with crucial differences. Like skeletal muscle, cardiac muscle is striated. It possesses those same light and dark bands because it too uses the same protein machinery to generate force. However, the shape of the cells is different. While skeletal fibers are long, separate cylinders, cardiac cells are short, fat, and branched. They look like tree branches or the roots of a plant, splitting and reconnecting with their neighbors. This branching architecture leads us to the most important feature of cardiac muscle, a feature found nowhere else: the intercalated disc. Imagine looking at the junction where two cardiac cells meet. It is not a smooth wall. Instead, it is a dark, jagged line that looks like a zipper or a step-like connection. This is the intercalated disc. It serves two vital functions. First, it acts as a mechanical anchor. holding the cells together so they don't rip apart when the heart beats. Second, and perhaps more poetically, these discs contain microscopic tunnels called gap junctions. These tunnels allow electrically charged ions to flow instantly from one cell to the next. Why is this important? It means that when one heart cell receives the signal to contract, it instantly passes that signal to its neighbor, who passes it to the next. The electrical wave sweeps through the heart so fast that the entire organ contracts as a single, unified entity. In physiology, we call this a functional syncytium. If skeletal muscles are a regiment of soldiers marching in line, cardiac muscle is a choir singing with one voice. So, when you visualize cardiac muscle, remember, it is striated like skeletal muscle, but it is involuntary. It is branched, and it is stitched together by intercalated discs, ensuring that the heart beats as one. Part 3. Smooth muscle. Finally, we move to the third and perhaps most mysterious family, smooth muscle. We find this tissue deep inside the body, lining the walls of your hollow organs. It wraps around your stomach, your intestines, and your bladder. It forms the walls of your blood vessels, the arteries and veins. It is found in the respiratory passages of your lungs. Like cardiac muscle, smooth muscle is involuntary. You cannot consciously command your stomach to digest your dinner faster, nor can you command your arteries to dilate. These muscles work silently in the background, managed by the wisdom of the autonomic nervous system. The name smooth muscle tells you exactly what it looks like. Under the microscope, it has no striations. There are no stripes. It appears smooth and uniform. This is because the internal proteins are not arranged in that rigid, geometric lattice we see in skeletal and cardiac tissue. They are arranged more randomly, spiraling through the cell. The cells themselves are distinct in shape. They are described as fusiform or spindle-shaped. Imagine a kayak, or a grain of rice wide in the middle and tapered to a point at both ends. Unlike the giant skeletal fibers, these are smaller cells, and they each contain a single, centrally located nucleus. The movement of smooth muscle is also unique. Skeletal muscle pulls quickly and powerfully. Cardiac muscle pumps rhythmically. But smooth muscle works in slow, sustained, wave-like contractions. Think of the way food moves through your body. It is not shot through like a cannon. It is pushed gently. We call this movement peristalsis. It is a rhythmic wave of contraction and relaxation that propels substances through the tubes of your body. Smooth muscle is built for endurance. It can hold a contraction like keeping a blood vessel clamped shut to maintain pressure for very long periods without using much energy and without tiring. It is the tortoise of the muscular system, slow, steady, and incredibly efficient. So, for smooth muscle, remember, it is not striated. It is involuntary. It is found in the walls of hollow organs, moving fluids and nutrients with a slow, tireless grace. Part 4. Comparison and Synthesis. Now, let us take a moment to step back and look at these three actors on the same stage. It is helpful to compare them side by side to let the knowledge settle. Think of control. Skeletal muscle is the only one that answers to your conscious will. Both cardiac and smooth muscles operate on the autopilot of the involuntary nervous system. This distinction allows you to focus on the world around you, while your biology takes care of the world within you. Think of speed. Skeletal muscle is the speed demon capable of rapid, explosive bursts of energy, but prone to fatigue. Cardiac muscle is the steady pacer, working at a consistent rhythm, never stopping, never tiring. Smooth muscle is the slow sustainer, working deliberately, efficiently, maintaining the tone of your internal systems. Think of appearance. Two are striated, skeletal, and cardiac. revealing their need for organized, forceful contraction. One is smooth muscle, revealing its need for flexibility and continuous squeezing. And think of nuclei. Skeletal muscle is the multinucleated giant. Cardiac and smooth muscles are content with single, or sometimes double, in cardiac nuclei, operating on a smaller cellular scale. It is also worth noting that while their primary job is motion, these muscles provide us with more than just movement. They provide us with heat. This is a function we often overlook. Muscle contraction requires energy, and as a byproduct of using that energy, heat is released. This is why you shiver when you are cold. Shivering is nothing more than your skeletal muscles rapidly contracting, not to move you, but to burn fuel and generate heat to warm your vital organs. In this way, your muscular system is also your internal furnace. Furthermore, they provide posture and stability. Even when you think you are standing still, your skeletal muscles are making tiny, imperceptible adjustments to keep you balanced against gravity. Without this constant, low-level tension called muscle tone, you would collapse. And internally, the sphincters of smooth muscle act as valves, holding fluids in the stomach or bladder until the appropriate time. They are the gatekeepers of your interior. We have covered a great deal of ground tonight. We have met the three families that comprise the muscular system. We have seen the skeletal muscle, the striated, voluntary soldier that moves your limbs and obeys your commands. We have seen the cardiac muscle, the striated, involuntary weaver of the heart, connected by intercalated discs to beat in unison. And we have seen the smooth muscle, the non-striated, involuntary linings of your organs, managing the slow flow of life's essential fluids. Take a moment to let that idea find its place. The complexity of your own design, the layers of cooperation happening right now. As you rest, your skeletal muscles are softening, letting go of the day's posture. Your smooth muscles are quietly digesting, regulating, flowing, and your cardiac muscle continues its steady, rhythmic vigil, a drum that requires no player. You now possess the foundational knowledge, the who is who of the muscular system. This understanding is the bedrock upon which we will build everything else. Having now met the families, we are prepared to look closer at the most prominent member of the group. We are ready to examine the architecture of strength. When we return for our next lesson, we will focus specifically on skeletal muscle. We will not look at the cells just yet, but at the gross anatomy, the layers of connective tissue that wrap, protect, and organize the muscle into the powerful cables that allow you to move the world. For now, release the concepts. You do not need to memorize the terms. Let the images of the striped fibers and the branching heart cells fade gently into the background. Trust that the logic has been planted. Rest your mind. We will pick up the thread again soon. Lesson 2. Gross Anatomy of Skeletal Muscle. Welcome back. It is good to have you here again. Take a moment to adjust your surroundings. Perhaps the room is quiet, the lights are low, and the demands of the day have finally receded into the distance. You are creating a space for yourself, a space for rest, and a space for gentle understanding. In our last session, we stood at a distance and observed the three great families of muscle tissue: skeletal, cardiac, and smooth. We learned to recognize them by their stripes and their shapes. But tonight, we are going to narrow our focus. We are going to zoom in on the specific tissue that allows you to interact with the world around you. We are turning our full attention to skeletal muscle. However, we are not yet ready to look through the microscope. Before we can appreciate the tiny molecular machines inside a cell, we must understand the grand architecture that holds it all together. This is the study of gross anatomy. The word gross here simply means large, or visible to the naked eye. It is what you would see if you held a muscle in your hand. When we think of a muscle, let's say, the biceps in your arm, we often imagine it as a solid block of red meat. But if we look closer, with the eye of an anatomist, we see that a muscle is not a single, solid block at all. It is a masterpiece of organization. It is a bundle of bundles, wrapped in layers of protective silk, designed to withstand incredible tension while remaining flexible. Tonight, we will peel back these layers, one by one, traveling from the outer surface deep into the interior structure. I want you to visualize a heavy-duty suspension cable, the kind that holds up a massive bridge. From a distance, that cable looks like one solid steel line, but if you were to cut it open and look at the cross-section, you would see that the large cable is actually made of smaller cables twisted together. And if you looked at one of those smaller cables, you would see it is made of even tinier wires. This structure, bundles within bundles, is exactly how your muscles are constructed. It is nature's way of building immense strength. Let us begin at the very surface. Imagine you are looking at a single, complete muscle. It is wrapped in a dense, tough outer coat called the epimysium. The word epimysium comes from Greek roots. Epi means upon or over, and mys refers to muscle. So, quite literally, this is the layer upon the muscle. This is not a delicate covering. It is a sheath of dense, irregular connective tissue. It needs to be tough because it serves as the primary container. It binds all the inner bundles together, preventing the muscle from bursting outward when it contracts. But the epimysium does more than just contain. It protects. It reduces friction. Your body is packed tight with muscles, bones, and organs. Your muscles slide against each other. The epimysium acts like a smooth jacket, allowing the muscle to glide against its neighbors without snagging or tearing. It separates the muscle from the surrounding environment, defining it as a distinct organ. Now, imagine we take a scalpel and gently slice through this outer epimysium. What do we find inside? We do not find a soup of cells. We find organization. We find the next level of bundling. Inside the muscle, the tissue is divided into compartments. These compartments are like the segments of a citrus fruit. Each of these bundles is called a fascicle. The word fascicle translates to a little bundle. If you have ever looked closely at cooked meat, like a pot roast or pulled pork, and seen how it shreds into stringy strips, you have seen fascicles. That grain of the meat is simply the alignment of these bundles. Each fascicle is wrapped in its own protective layer of connective tissue called the perimysium. Peri means around, so this is the tissue around the muscle bundles. The perimysium is slightly more fibrous and elastic than the outer coat. It surrounds the fascicle like a sleeve. This level of organization is crucial for control. Your brain does not always need to activate the entire muscle at once. Sometimes you need a gentle touch. Sometimes you need a powerful lift. By organizing muscle fibers into these fascicles, the nervous system can selectively activate just a few bundles for a delicate movement, or recruit all of them for a maximum effort. The perimysium provides the structural borders for these functional groups. So, we have the outer epimysium. Inside that, we have the perimysium wrapping the fascicles. Now, let us go deeper still. Imagine we pluck a single fascicle out of the muscle, like pulling a single segment from an orange. If we look inside this fascicle, we finally find the individual muscle cells. In the world of anatomy, we call these cells muscle fibers. We call them fibers because they are incredibly long and thin. Some of them run the entire length of the muscle, spanning from hip to knee. They are the actual living units that do the work of contraction. But even here, at the level of the single cell, nature is not done with its wrapping. Each individual muscle fiber is surrounded by a delicate, wispy layer of connective tissue called the endomysium. Endo means within. This is the deepest layer. The endomysium is very different from the tough outer coats. It is made of fine reticular fibers. It is soft and fragile. Its job is to electrically insulate each muscle cell from its neighbors. This ensures that the electrical signal to contract doesn't accidentally jump to the wrong cell. It also creates a microscopic space for the tiniest blood capillaries and nerve endings to reach every single cell. It is the intimate interface between the muscle fiber and the life-giving fluids of the body. Let's pause and visualize this hierarchy again, building it from the inside out. First, you have the single muscle fiber wrapped in the delicate endomysium. Group a bunch of these fibers together, and you get a fascicle wrapped in the fibrous perimysium. Group a bunch of fascicles together, and you get the whole muscle wrapped in the tough epimysium. It is a tube within a tube within a tube. Now, you might be asking, why so much wrapping? Why so much connective tissue? Why not just pack the cells together like sardines? The answer lies in the transmission of force. This is one of the most beautiful mechanics of the body. When a muscle fiber contracts, it pulls on its own wall, the endomysium. The endomysium pulls on the perimysium. The perimysium pulls on the epimysium. All of these connective tissue layers are continuous with one another. They are glued together. And here is the magic. At the ends of the muscle cells themselves stop, but the connective tissues do not. The endomysium, perimysium, and epimysium all extend beyond the muscle fibers. They merge together, twisting and thickening to form a new structure, the tendon. So, a tendon is not a separate thing glued to the end of a muscle. A tendon is the connective tissue layers of the muscle gathered together into a rope. It is the anchor. This tendon then travels to the bone. But it doesn't just stick to the surface. It weaves right into the covering of the bone, called the periosteum. It literally buries itself into the mineral matrix. This creates an unbroken chain of physical connection. When the tiny cells inside the muscle shorten, they pull on their wrappers, which pull on the tendon, which pulls on the bone. Without these layers of connective tissue, the force generated by the cells would be useless, like an engine revving without a transmission. These layers harness the power and deliver it to the skeleton. Sometimes, this attachment is not a rope-like tendon. In some parts of the body, like the abdomen or the top of the skull, the connective tissues flatten out into a broad, flat sheet. This sheet is called an aponeurosis. It functions exactly like a tendon, but instead of focusing force on a single point, it spreads the force over a wide area. It is a sheet of white, pearly tissue, glistening and strong. We must also briefly mention the deep fascia. If the epimysium is the coat on the muscle, the deep fascia is the coat on the entire limb. It is a dense sheet of connective tissue that wraps around groups of muscles. For example, it groups all the quadriceps muscles together in the thigh, separating them from the hamstring muscles in the back. It acts like a compression stocking, keeping the muscle groups compact and aiding in the return of blood to the heart. Speaking of blood, we must not forget that a muscle is a living organ. It is not just a mechanical pulley, it is voracious for energy. If you were to trace the path of the connective tissues we just discussed, you would find that they serve as a highway system. Arteries, veins, and nerves ride along these connective tissues to reach the deep interior of the muscle. Every skeletal muscle is supplied by one main nerve, one artery, and one or more veins. These vessels enter the center of the muscle and then branch out, following the perimysium into the fascicles, and finally branching into tiny capillaries in the endomysium. This blood supply is critical. Muscle contraction requires huge amounts of energy and oxygen. The blood brings the fuel, the glucose and oxygen, and carries away the waste products, like carbon dioxide and lactic acid. This is why the muscle is so red. It is rich with blood and iron. Furthermore, the nerve supply is equally vital. Skeletal muscle, as we learned, is voluntary. It does nothing without a command. Every single muscle fiber, and remember, there are thousands or millions in a single muscle, must have a nerve ending connected to it. The nerve must penetrate the epimysium, travel through the perimysium, and reach the endomysium to deliver its spark to the cell. It is a hardwired connection, physically linking your nervous system to your muscular system. So, as we synthesize this lesson, I want you to appreciate the complexity of what looks like a simple piece of anatomy. Think of the resilience of this design. The connective tissue layers provide elasticity. When you stretch your arm, you are stretching these sheaths. When you release, they recoil, helping the muscle return to its resting shape. They provide a passive springiness that protects the delicate cells from tearing apart. Think of the protection. The delicate machinery of the cell is cushioned by 3 layers of shock absorption. And think of the unity. These layers turn millions of microscopic individual contractions into a single, unified movement of a limb. It turns the microscopic whisper of a cell into the shout of physical strength. As we close this session, try to feel the weight of your own limbs resting on the bed. Visualize the muscles inside them, not as blocks of meat, but as intricate cables. See the epimysium, the silver wrapping, holding everything together. See the perimysium, dividing the interior into organized bundles. See the endomysium, gently cradling each sleeping cell. And see the tendons, the strong ropes anchoring this potential energy to your bones. It is a quiet, sturdy architecture, waiting for your command, but currently at rest. You have now grasped the gross anatomy, the big picture structure of the muscle. You understand how the force is harnessed and transmitted. But the true mystery lies deeper. The actual engine, the machinery that generates the force, is hidden inside the muscle fiber itself. In our next lesson, we will shrink ourselves down. We will pass through the endomysium and enter the cell. We will explore the microscopic anatomy of the muscle fiber. We will discover a world of filaments, of sliding bridges, and of molecular geometry that rivals any machine humans have ever built. For now, let the layers protect you. Let the structure support you. Rest your thoughts and drift into the quiet space between the lessons. Lesson 3: Microscopic Anatomy of the Muscle Fiber. Welcome back. I hope you have found a place of stillness. Let your breathing slow down, matching the rhythm of these words. In our last session, we were builders. We looked at the muscle as a whole, a heavy cable wrapped in layers of silver skin, attaching bone to bone. We saw the gross anatomy, the structures large enough to be held in the hand. Tonight, we change our perspective entirely. We are going to leave the world of the visible and descend into the world of the microscopic. We are going to shrink ourselves down, passing through the outer epimysium, navigating through the fascicles, and slipping through the delicate endomysium until we stand before a single muscle cell. In the language of anatomy, this cell is called a muscle fiber. It is the fundamental unit of your physical strength. And while it is called a cell, it is unlike any other cell in your body. It is a masterpiece of biological engineering, a machine designed with one singular purpose: to shorten, to pull. Tonight, we will open the hood of this machine and examine the engine. Imagine, if you will, that we are floating just outside this muscle fiber. The first thing you notice is its size. Most human cells are tiny, invisible specks, but a muscle fiber is immense. It is a long, cylindrical tube that can be up to 30 centimeters long, running the entire length of your thigh muscle. It is a giant. This giant is enclosed in a plasma membrane, but because this is muscle, we give it a special name. We call it the sarcolemma. The prefix sarco comes from the Greek word for flesh. You will hear this prefix often tonight. The sarcolemma is the skin of the cell. But it is not just a passive wrapper, it is an electrically active fence. It is capable of conducting an electrical wave across its surface, just like a wire. This ability to conduct electricity is vital, for it is the spark that will eventually ignite the contraction. Just beneath this surface, just under the sarcolemma, we see the nuclei. Remember, we learned that skeletal muscle fibers are multinucleated. You can see them pressed against the outer wall, dozens or even hundreds of them scattered along the length of the fiber. They are pushed to the periphery because the center of the cell is packed so tight with machinery that there is simply no room for them in the middle. Let us pass through the sarcolemma and enter the interior. We are now swimming in the fluid of the cell. In a normal cell, this is called cytoplasm. Here, in the muscle, we call it sarcoplasm. The sarcoplasm is rich and gelatinous. It contains large amounts of glycosomes, which are granules of stored glycogen, sugar, fuel, waiting to be burned. It also contains a red protein called myoglobin. Myoglobin is a cousin to the hemoglobin in your blood. Its job is to grab oxygen and hold onto it, creating a local oxygen reserve right here inside the cell, so the muscle doesn't suffocate during intense work. But the sarcoplasm is not an empty swimming pool. As we look around the interior of this vast cell, we see that it is dominated by long, rod-like structures. These rods run parallel to each other, extending the entire length of the cell, packed together like logs in a truck or drinking straws in a box. These rods are called myofibrils. Please do not confuse the muscle fiber with the myofibril. The muscle fiber is the cell itself. The myofibrils are the long tubes inside the cell. A single muscle fiber might contain hundreds to thousands of these myofibrils. They are so densely packed that they account for about 80% of the cell's volume. They squeeze the organelles, the nuclei, and the fluid to the edges. These myofibrils are the actual contractile elements. They are the engines. If we look closely at a single myofibril, we notice something beautiful. These bands line up perfectly with the bands on the neighboring myofibrils, which is what gives the entire muscle fiber its striated appearance under a microscope. The dark bands are called A bands. The light bands are called I bands. Why the stripes? The answer lies in the arrangement of the tiniest structures of all. We must zoom in even further. We are going to enter the myofibril itself. Inside the myofibril, we find the functional unit of the muscle. This unit is called the sarcomere. The sarcomere is the smallest segment of muscle contraction. Imagine a long train made of identical boxcars. The myofibril is the train. The sarcomere is a single boxcar. The myofibril is simply a chain of thousands of sarcomeres linked end to end. Let us map the geography of one sarcomere. At each end of the sarcomere, there is a zigzagging line of protein. We call this the Z-disc or Z-line. The Z-disc forms the boundary. A sarcomere is defined as the region between two Z-discs. When a muscle contracts, what is actually happening is that these Z-discs are being pulled closer together. The entire boxcar shortens, and because thousands of boxcars shorten at the same time, the entire fiber shortens. But what pulls the Z-discs? What is the force inside the boxcar? Floating between the Z-discs are thousands of tiny protein strands called myofilaments. There are two types you must know. The relationship between these two is the love story of movement. There is the thick filament and there is the thin filament. Let us introduce the thick filament first. This filament is located in the center of the sarcomere. It is made primarily of a protein called myosin. Visualize a golf club. It has a long, straight shaft and a knobby head. Now, imagine bundling 300 golf clubs together, with the shafts in the middle and the heads sticking out at both ends. Spiraling around the bundle, This is the thick filament. Those knobby heads are called the myosin heads. They are the workers. They are the tiny hands that will eventually reach out, grab the thin filament, and pull. They are chemically tuned to pivot, like the hinge of a door. This pivoting action is the literal stroke of movement. Now, let us introduce the partner, the thin filament. The thin filament is anchored to the Z-disc and extends toward the center of the sarcomere, overlapping with the thick filaments. It is made primarily of a protein called actin. If the myosin is a bundle of golf clubs, actin looks like a twisted double strand of pearls. Each pearl is a globular actin molecule, and on each pearl there is a specific spot, a binding site, that is perfectly shaped for the myosin head to grab onto. So, the stage is set. The myosin heads are hovering in the center. The actin strands are hanging near them. The myosin wants to grab the actin. It is chemically desperate to grab the actin. If they touch, the myosin will pull. The Z-discs will come together, and you will move. But wait, we cannot have our muscles contracting uncontrollably all the time. We need a way to turn this system off. This brings us to the regulatory proteins. These are the guardians of the actin. Wrapped around the actin pearls is a long, rod-shaped protein called tropomyosin. In a relaxed muscle, the tropomyosin acts like a shield. It spirals around the actin and physically covers those binding sites. It blocks the myosin heads from making contact. It says, "No, not yet." Sitting on top of the tropomyosin is another smaller protein complex called troponin. Think of troponin as the lock on the door. It holds the tropomyosin in place. It is the gatekeeper. So, in a resting muscle like yours right now, the machinery is paused. The myosin heads are cocked and ready. The actin is present. But the tropomyosin is blocking the connection, held fast by the troponin. The key to unlocking this door, as we will learn later, is calcium. When calcium arrives, it binds to troponin, which moves the tropomyosin aside, and the dance begins. We have almost completed our tour of the cell, but there is one more critical system we must visualize. We have the engine, the sarcomeres, and we have the fuel, the mitochondria and glycogen. But how does the command to contract reach all these deep sarcomeres at the exact same moment? The muscle fiber is thick. If the electrical signal stayed only on the outside skin, the sarcolemma, the sarcomeres in the center of the cell would never know it was time to move. To solve this, the muscle fiber has an incredible plumbing system. First, there are the T-tubules, transverse tubules. Imagine pushing your finger into a soft balloon. The rubber stretches inward, creating a tunnel. The T-tubules are essentially tunnels of the outer sarcolemma that dive deep into the center of the cell. They run like subway tunnels perpendicular to the myofibrils. Because they are continuous with the surface, when an electrical signal sweeps across the skin of the cell, it also races down these tunnels, bringing the spark of electricity deep into the core of the fiber. Second, there is the sarcoplasmic reticulum, SR. This is a complex network of smooth, fluid-filled channels that surround each myofibril like a lacy, crocheted sleeve. It fits loosely around the sarcomeres. The sarcoplasmic reticulum has one main job: to store calcium. It works tirelessly to pump calcium ions out of the sarcoplasm and store them safely inside its tubes. It is a reservoir. At the point where a T-tubule dives down, the sarcoplasmic reticulum widens into large chambers called terminal cisterns. These three structures, the T-tubule in the middle, sandwiched between two terminal cisterns of the SR, form a structure called a triad. The triad is the critical junction. It is where the electricity meets the chemistry. The electrical signal comes down the T-tubule subway. It hits the triad. This triggers the terminal cisterns to open their floodgates. Calcium pours out of the sarcoplasmic reticulum and floods the sarcomere. This calcium finds the troponin, the lock, opens the door, and allows the myosin to grab the actin. So, let us synthesize this complex world we have just entered. Visualize the muscle fiber, the giant cell. Inside, it is packed with rod-like myofibrils. The myofibrils are chains of sarcomeres. The sarcomeres are battlegrounds of thick myosin and thin actin filaments. The actin is guarded by troponin and tropomyosin. Surrounding all of this is the lacy sarcoplasmic reticulum holding the calcium key, and piercing through it all are the T-tubules waiting to carry the command. It is a machine of breathtaking complexity, yet it operates with silent, seamless efficiency. Every time you blink, every time you breathe, billions of these tiny sarcomeres are sliding past each other, locking and unlocking, fueled by the rush of ions and the burning of sugar. As you lie here, resting, your muscles are in a state of quiet readiness. The calcium is safely stored away in the reticulum. The tropomyosin shields are up. The myosin heads are resting. The machine is idling, waiting for the next signal. You have now visualized the stage where the action takes place. You know the players: actin, myosin, calcium, and the membranes. But the machine does not start itself. It requires a pilot. It requires a signal from the nervous system to begin the cascade. In our next lesson, we will travel to the surface of the muscle fiber, to the specific point where the nerve meets the muscle. We will explore the neuromuscular junction. We will see how a thought in your brain is translated into a chemical letter that crosses the gap to wake the sleeping giant. For now, let the image of the lacy networks and the pearl strands fade. Let the complexity dissolve into the simple feeling of relaxation in your own limbs. You are built of remarkable things. Rest in that knowledge. Lesson 4. The Neuromuscular Junction. Welcome back. Take a deep, slow breath and let it go. With every exhalation, allow yourself to sink a little deeper into the surface that supports you. You are safe, you are resting, and you are ready to continue our exploration of the human body. In our previous sessions, we have been explorers of structure. We have mapped the gross anatomy of the muscles, and we have dived deep into the microscopic world of the muscle fiber to see the filaments of actin and myosin waiting in silence. We know the machine is built. We know the engine is assembled. But a machine, no matter how perfect, cannot start itself. It requires a spark. It requires a command. Tonight, we stand at a fascinating border crossing. We are going to explore the connection between the two master systems of the body, the nervous system, which commands, and the muscular system, which obeys. This meeting point has a name. It is called the neuromuscular junction. It is a place of translation. On one side, we have the language of the brain electricity racing down a wire. On the other side, we have the language of the muscle mechanical force and movement. The neuromuscular junction is the interpreter. It is the exact spot where a thought becomes a physical action, where the intangible will to move becomes the tangible reality of motion. Let us visualize the geography of this place. Imagine a single nerve cell, known as a motor neuron. Its cell body sits safely within your spinal cord or brainstem, but it sends out a long, wire-like extension called an axon. This axon travels a great distance, sometimes 2 or 3 feet, weaving through your body to reach a specific muscle. As the axon approaches the muscle, it branches out. It splits into tiny terminals, like the roots of a tree. Each of these tiny roots reaches out to touch a single muscle fiber. But here is the most remarkable detail: they do not actually touch. If you were to look at this junction under the most powerful microscope, you would see a tiny, infinitesimal gap between the end of the nerve and the surface of the muscle. This gap is called the synaptic cleft. It is a chasm, filled with a gel-like substance rich in glycoproteins. It separates the two worlds. The nerve is on one side, the muscle is on the other. This creates a problem. The electrical signal traveling down the nerve cannot jump through empty space. Electricity needs a wire. It cannot simply fly across the gap. So, how does the command get across? The body must switch methods. It must change from an electrical signal to a chemical one. It is like a runner reaching a river. They must stop running, get into a boat to cross the water, and then start running again on the other side. Let us slow down the clock and watch this event happen, millisecond by millisecond. We begin on the nerve side. This area is called the axon terminal. Inside this terminal, floating in the fluid, are thousands of tiny bubbles. These bubbles are called synaptic vesicles. Think of them as cargo ships. Inside each bubble is a precious cargo, a specific chemical messenger or neurotransmitter called acetylcholine. We often abbreviate this as HA. Acetylcholine is the letter that contains the message contract. Thousands of these letters are packed into the vesicles, waiting near the edge of the river, waiting for the signal to launch. Suddenly, the signal arrives. A wave of electricity, called an action potential, races down the axon. It reaches the terminal. This electrical arrival changes the voltage of the membrane. This change in voltage opens special gates in the nerve wall. These are called voltage-gated calcium channels. When these gates open, calcium, which is plentiful outside the nerve, rushes inside. We have seen calcium before, and we will see it again. In the body, calcium is almost always the signal to go. When calcium floods into the nerve terminal, it acts as a trigger. It tells the synaptic vesicles, those little cargo ships, to move. They rush toward the very edge of the nerve membrane. They fuse with the outer wall and burst open, spilling their contents into the gap. This process is called exocytosis. Thousands of molecules of acetylcholine flood into the synaptic cleft. They are released from the safety of the nerve and are now drifting across the river. Now, let us shift our perspective to the other side of the gap. We are standing on the surface of the muscle fiber. The part of the muscle membrane that sits directly across from the nerve is specialized. It is called the motor end plate. It is not smooth. It is highly folded, like a crumpled piece of velvet. These folds, called junctional folds, serve a very specific purpose. They increase the surface area. A larger surface means more room for receptors. And indeed, sitting on these folds are millions of acetylcholine receptors. These receptors are chemically gated ion channels. Think of them as locked doors waiting for a specific key. They are shut tight, preventing anything from entering the muscle. As the acetylcholine drifts across the cleft, it finds these receptors. It fits perfectly into the binding site. The key turns the lock. Boom! The doors fly open. These are not just any doors. They are channels for sodium and potassium. When the channel opens, sodium, Na+, which is highly concentrated outside the cell, rushes in. At the same time, potassium, K+, which is concentrated inside, trickles out. However, the rush of sodium entering is much stronger than the trickle of potassium leaving. This influx of positively charged sodium changes the electrical state of the muscle interior. The inside of the cell, which is normally negatively charged, suddenly becomes more positive. This local shift in voltage is called the end plate potential. It is the spark that has successfully jumped the river. The chemical messenger, acetylcholine, has done its job. It has opened the door, and the electricity has been recreated on the muscle side. Now, a chain reaction begins. This local spark at the motor end plate triggers voltage-gated channels nearby on the sarcolemma, the muscle skin. A new action potential is born. This wave of electricity sweeps away from the junction, racing across the surface of the muscle fiber in all directions, like ripples in a pond after a stone is thrown. It is this wave that will eventually dive deep into the cell to trigger the contraction we discussed earlier. But we must pause here. We have a loose end. What happens to the acetylcholine? We cannot leave it floating in the gap. If we did, it would keep binding to the receptors again and again. The doors would stay open. The muscle would stay contracted forever in a permanent spasm. We need the signal to be brief. We need to be able to stop the movement as quickly as we started it. Nature has a solution. It is a cleaning crew. Living inside the synaptic cleft, embedded in the fibers of the gap, is an enzyme called acetylcholinesterase. Its name is a mouthful, but its job is simple. It is a breaker. Almost as soon as acetylcholine is released, this enzyme hunts it down. It breaks the acetylcholine into two parts, acetic acid and choline. It effectively tears up the letter. Once the acetylcholine is broken, it can no longer fit in the receptor lock. The ion channels slam shut. The flow of sodium stops. The muscle membrane returns to its resting state, ready for the next signal. This cleanup happens with incredible speed within a fraction of a millisecond. It ensures that one nerve impulse produces exactly one muscle contraction. It allows for precision. It allows you to tap your finger once, rather than having it lock up in a cramp. The choline part of the broken molecule is not wasted. It is taken back up by the nerve terminal, recycled, and used to build new acetylcholine for the next message. The body is always economical. Let us take a moment to reflect on the vulnerability of this system. Because the neuromuscular junction relies on chemistry, it is a point of weakness. It can be interfered with. For example, certain toxins, like curare, used on the tips of arrows by indigenous hunters in South America, work by blocking the receptors on the muscle. The acetylcholine is released, but it has nowhere to sit. The letter is delivered, but the mailbox is nailed shut. The result is paralysis. The muscle never hears the command. On the other hand, some nerve gases work by inhibiting the cleanup crew, the acetylcholinesterase. If the cleaner doesn't work, the acetylcholine stays in the gap, and the muscle contracts violently and cannot relax. I tell you this not to worry you, but to illustrate the exquisite balance your body maintains every second of your life. Right now, as you lie in bed, your muscles are relaxed because no acetylcholine is being released. The gates are closed. The nerve is silent, but if you were to decide to move your finger, the system would spring into action. The electrical wave, the calcium entry, the vesicle fusion, the release across the cleft, the binding to the receptor, the rush of sodium, the breakdown by the enzyme. All of this happens in less time than it takes to blink. It is a relay race, passed from electricity to chemistry and back to electricity. Let us revisualize the entire sequence as a flowing narrative, letting the technical terms soften into the story. Imagine the long, dark tunnel of the nerve. A spark of light travels down it, the intention of your mind. It reaches the end of the tunnel. The light fades, but it triggers the opening of the cargo bay doors. Thousands of tiny boats, the acetylcholine, launch into the narrow river. They drift across the dark water of the synaptic cleft. On the other bank, the shore of the muscle, there are thousands of landing docks, the receptors. The boats dock. The gates of the city fly open. Energy floods into the city. A new fire is lit on the shores of the muscle, and just as quickly, the boats are dissolved, cleared away by the waters, leaving the dock empty and ready for the next arrival. This is the neuromuscular junction. It is the handshake between the brain and the body. It is the bridge over which your will travels to become action. This process, the successful transmission of the signal, creates the electrical wave on the muscle surface. This wave is called excitation. But excitation is only the first half of the story. The muscle is now electrically excited, but it hasn't physically moved yet. The filaments inside haven't slid. We have the spark, but we do not yet have the fire. How does this electrical wave on the surface get deep inside the cell to the actin and myosin we met in the last lesson? How does it unlock the calcium from the sarcoplasmic reticulum? This next step is the link between the electricity and the movement. It is called Excitation-Contraction Coupling. It is the mechanism that connects the command to the work. It is the gears turning after the key has been turned. In our next lesson, we will follow that electrical wave as it dives down the T-tubules and opens the floodgates of the cell. We will watch the chemistry of the junction transform into the physics of the pull. For now, rest your mind on the image of the bridge, the gap that is not a barrier, but a point of connection. Your body is a continuous conversation, whispers passing across the clefts, keeping you whole, keeping you moving, and tonight, allowing you to rest. Let the river flow quietly, let the gates close, let the silence return. Lesson 5. Excitation, Contraction, Coupling. Welcome back. Let your body find its natural heaviness. Allow your shoulders to drop away from your ears and let your hands uncurl. You are here, in this quiet space, to learn about the incredible machinery that allows you to be here at all. In our last session, we stood at the banks of the neuromuscular junction. We watched as the nerve cell released its chemical messenger, acetylcholine, across the gap. We saw the muscle receive the letter. We saw the spark ignite. An electrical wave, a ripple of energy called an action potential, began to race across the surface of the muscle fiber. But here is the puzzle we must solve tonight. The muscle fiber is a giant cell. It is thick and dense. The machinery that actually does the pulling the actin and myosin filaments we met earlier is buried deep inside the core of this cell, far away from the surface skin where the electricity is flowing. Having electricity on the surface is not enough. It is like having a wire running along the outside of a house when the light bulb is in the basement. We need to get the power to the bulb. We need to connect the electrical excitation on the surface to the mechanical contraction in the deep interior. This process, the bridge between the spark and the movement, is called excitation-contraction coupling. It is perhaps the most sophisticated sequence of events in muscle physiology. It is the story of how a silent chemical key unlocks a flood. Let us visualize the geography again. Imagine you are riding that wave of electricity sweeping across the surface of the muscle. You are moving fast. Suddenly, the floor drops out from under you. You plunge down a deep, dark tunnel. These tunnels are called transverse tubules, or simply T-tubules. We mentioned them briefly before, but now we must truly understand them. The T-tubules are not separate structures. They are continuations of the outer membrane. Imagine poking your finger into a soft loaf of dough. The dough surrounds your finger, but the skin of the dough has now gone deep inside the loaf. That is a T-tubule. It is a hollow tube of surface membrane that dives straight down into the center of the muscle fiber, winding its way around the myofibrils. Because the T-tubule is made of the same material as the surface, it can carry the same electricity. The action potential does not stop at the surface. It races down these tunnels, bringing the electrical command into the deepest, darkest parts of the cell. It ensures that the myofibrils in the center of your thigh muscle get the message at the exact same instant as the ones on the outside. This plumbing system is what allows a thick muscle to contract as a single, unified block. But where do these tunnels lead? They lead to the vault. Wrapping around each myofibril, like a lacy, fluid-filled sleeve, is the sarcoplasmic reticulum. We often call it the SR. The SR is a storage tank. And what does it store? It stores calcium. I want you to think of calcium not as a bone builder, but as a dangerous, high-energy signal. In the cytoplasm of a healthy cell, calcium levels are kept incredibly low. Why? Because calcium is a trigger. It changes things. It turns enzymes on and off. If calcium were allowed to float freely all the time, the cell would be in a state of constant, chaotic hyperactivity. So, the muscle cell works tirelessly to pump calcium out of the fluid and lock it away inside the sarcoplasmic reticulum. It is like a dam holding back a massive river. Inside the SR, the concentration of calcium is thousands of times higher than outside. It is pressurized. It is waiting to burst out. Now, look closely at where the T-tubule meets the sarcoplasmic reticulum. The T-tubule runs right next to the bulging ends of the SR, which we call the terminal cisterns. Together, they form a structure called a triad. One T-tubule sandwiched between two terminal cisterns. This triad is the magic switch. Embedded in the wall of the T-tubule is a voltage-sensitive protein. Think of it as a motion detector. It is sensitive to electricity. Embedded in the wall of the SR, right next to it, is a calcium release channel. Think of this as the floodgate of the dam. In skeletal muscle, these two proteins are physically linked. They touch each other. Here is the sequence, happening in a fraction of a millisecond. The electrical wave races down the T-tubule. The voltage-sensitive protein detects the electricity. It changes shape. It twists. Because it is physically connected to the floodgate on the SR, when it twists, it pulls the gate open. It is mechanical. It is like pulling the plug in a bathtub. The moment the gate opens, the calcium, which has been packed tight inside the SR, explodes out into the cell fluid. It floods the sarcomeres. It washes over the actin and myosin filaments. This is the coupling in excitation-contraction coupling. The electrical excitation has successfully triggered the release of the chemical trigger. But what does the calcium do? Why is it the key? To understand this, we must revisit our old friends, the thin filaments. Recall that the actin filament is like a twisted strand of pearls. Each pearl has a binding site, a spot where the myosin wants to grab it. But in a resting muscle, that spot is covered. A long, stringy protein, called tropomyosin, lies along the actin strand, blocking the binding sites. It is like a piece of tape over a keyhole. Holding this tape in place is another protein, a small complex called troponin. Troponin is the lock, and it has a very specific shape. It has a notch that is perfectly designed to catch one thing: a calcium ion. So, here comes the flood. The calcium ions rush out of the SR and bind to the troponin. When calcium binds to troponin, the troponin changes shape. It twists. Because troponin is holding onto the tropomyosin tape, when it twists, it pulls the tropomyosin away. Imagine a guard standing in front of a door. Calcium pays the guard. The guard steps aside and opens the door. Suddenly, the binding sites on the actin are exposed. The tape is gone. The myosin heads, which have been hovering nearby, cocked and ready with energy, see their chance. They instantly reach up and bind to the exposed actin. This physical connection, the myosin head attaching to the actin filament, is called a cross bridge. At this exact moment, the potential becomes kinetic. The coupling is complete. The electrical signal has led to the release of calcium. The calcium has moved the guard, and the muscle proteins have linked hands. The engine is engaged. The actual movement, the power stroke, where the myosin pulls the actin, will be the subject of our next lesson. But tonight, we must appreciate the control system. We must appreciate how the body ensures this doesn't go on forever. You see, muscle contraction is a cycle of on and off. If the calcium stayed in the fluid, the guards would stay away, the doors would stay open, and the muscle would cramp into a permanent, rigid contraction. We need a way to stop. This is where the energy cost of relaxation comes in. We often think of exercise as burning energy and resting as free. But chemically, relaxation is expensive. Embedded in the membrane of the sarcoplasmic reticulum are active transport pumps. They are the vacuum cleaners of the cell. The moment the floodgates open and calcium rushes out, these pumps are already working to suck it back in. They use ATP, cellular energy, to grab calcium ions and force them back into the storage tank against the flow. It is a race. As long as the nerve is firing, the floodgates stay open and calcium continues to pour out faster than it can be pumped back in. The muscle stays contracted, but the moment the nerve stops firing, the electrical signal on the T-tubule dies. The voltage sensor twists back. The floodgate slams shut. Now, the pumps gain the upper hand. They rapidly vacuum up the remaining calcium. As the calcium levels in the fluid drop, the calcium ions detach from the troponin. Without the calcium, the troponin returns to its original shape. It pushes the tropomyosin shield back over the binding sites. The door is locked again. The myosin has nothing to grab. It lets go. The muscle fiber relaxes and returns to its resting length. This is why rigor mortis happens after death. When we die, our cells stop making ATP, energy. Calcium leaks out of the storage tank and cannot be put back. It binds to troponin, the guards step aside, and the muscles contract. But because there is no ATP to help the myosin let go, a detail we will cover next time, or to pump the calcium back, the muscles lock in a permanent stiffness. It is a somber reminder that relaxation is an active, living process. It requires energy to keep the calcium hidden away. Let us take a moment to integrate this complex dance into a single, flowing mental movie. Visualize the action potential, the spark racing along the surface of the muscle. See it dive down the T-tubules, plunging deep into the interior. See it trigger the voltage sensor, which yanks open the gates of the sarcoplasmic reticulum. Hear the rush of the calcium flood, pouring out like water from a dam. Watch the tiny troponin molecules catch the calcium and twist. See the long tropomyosin shield slide away, revealing the binding sites on the actin. And finally, see the myosin heads reach up and grab hold. Snap. Connection. The bridge is built. This is excitation-contraction coupling. It is the translation of a code. The code of the nervous system is frequency and voltage. The code of the muscular system is force and tension. Calcium is the translator that speaks both languages. It is fascinating to realize that the difference between a clumsy movement and a graceful one, or a weak effort and a strong one, is often just a matter of how much calcium is released and how fast. When you train your muscles, you are not just building bigger fibers. You are training this system to be more efficient, to release and recapture this chemical signal with greater speed and precision. As you lie there now, your calcium is safely locked away. Your SR pumps have done their job. Your tropomyosin shields are in place. Your muscles are soft, pliable, and at rest. The machinery is silent, but it is ready. There is a profound comfort in knowing that your body possesses such intricate safeguards. It does not move without cause. It requires a key. It requires energy to engage and energy to disengage. It is a system built for control. We have now reached the critical threshold. The myosin has grabbed the actin. The cross bridge is formed. But now, the real work begins. The myosin must pull. The sarcomere must shorten. The muscle must contract. In our next lesson, we will watch the mechanical cycle itself. We will study the sliding filament mechanism. We will see the myosin heads nod and pull, hand over hand, in a microscopic tug-of-war that generates the power to lift a mountain or cradle a child. For now, let the floodgates close in your mind. Let the pumps finish their work. Let the calm return to the cell. Lesson 6. The sliding filament mechanism. Welcome back. Settle yourself into the comfort of this moment. Let the world outside these walls fade away. You are safe, you are still, and you are ready to witness the culmination of everything we have discussed so far. In our previous lessons, we have meticulously built the stage. We know the actors, the thick myosin filaments and the thin actin filaments. We know the trigger, the flood of calcium that unlocks the doors. We know the energy source, the ATP waiting in the wings. We have brought the nerve signal to the muscle, sent it deep into the tubules, and released the calcium to expose the binding sites. Everything is ready. The key has turned. The door is open. Now, we watch the movement itself. Tonight, we explore the sliding filament mechanism. This is the physics of life. It is the explanation for how a collection of microscopic proteins can generate the force to lift a heavy box, to sprint across a field, or to simply draw a breath while you sleep. It is a story of attachment, pulling, releasing, and resetting a cycle that repeats billions of times in the blink of an eye. For a long time, scientists were baffled by muscle contraction. In the early days of biology, it was believed that protein filaments worked like springs or accordions, that they physically crinkled up and folded to become shorter. It made sense intuitively. If a muscle gets shorter, the things inside it must get shorter too, right? But in the 1950 seconds, two independent teams of researchers, both coincidentally named Huxley, proposed a radical new idea. They suggested that the proteins do not shorten, the actin doesn't curl up, the myosin doesn't shrink. Instead, they remain exactly the same length. They simply slide past one another. Imagine two telescopes sliding into each other. The tubes don't get smaller, the amount of overlap changes. Or imagine clasping your hands together and sliding your fingers between each other. Your fingers don't shrink, but the total width of your hands decreases. This is the sliding filament theory. It changed our understanding of biology forever. To see this happen, we must zoom in to the single sarcomere, that boxcar unit of the muscle we visited in Lesson 3. Remember the geography. The myosin is the thick bundle in the center. The actin is the thin strand attached to the walls, the Z-discs. The goal of contraction is to pull those walls closer together. The myosin heads are the workers. They are hovering in the center, waiting to grab the ropes of actin and pull them inward. This process occurs in a four-step loop known as the cross-bridge cycle. Let us slow down time and watch a single cycle unfold, step by step, with the precision of a watchmaker. Step 1. Cross-bridge formation. At this moment, the calcium levels are high. The tropomyosin guards have moved aside. The binding sites on the actin pearl strand are naked and exposed. The myosin head is already energized. It is cocked like the hammer of a gun or a set mousetrap. It is holding stored energy. Chemical attraction takes over. The myosin head reaches up and physically attaches to the actin binding site. This connection is profound. It is the moment potential energy becomes a physical bridge. The myosin and actin are no longer separate entities. They are linked. This link is called the cross bridge. Step 2. The power stroke. This is the main event. This is where the movement happens. When the myosin head first attaches, it is holding onto two small molecules, ADP, adenosine diphosphate, and inorganic phosphate. These are the exhaust fumes of the energy it used to get ready. The moment the cross-bridge forms, the myosin releases these molecules. It lets them go. This release triggers a shape change. The myosin head, which was stretched out, suddenly snaps or pivots toward the center of the sarcomere. Think of it like a rower in a boat, pulling the oar back. Or think of flexing your wrist. Because the myosin is stuck to the actin, This pivot pulls the actin filament along with it. It drags the thin filament toward the center line, the M line. This pivot is called the power stroke. It is a tiny movement, only about 10 nanometers, but it is powerful. It pulls the Z-disc slightly closer to the center. The muscle shortens, just a fraction. Step 3. Cross-bridge detachment. Now we have a problem. The myosin head has pivoted. It has pulled the rope, but it is still stuck to the actin. It is like a rower who has finished the stroke but hasn't taken the oar out of the water. If it doesn't let go, it can't pull again. To break this bond, we need a new player. We need fresh energy. A molecule of ATP, adenosine triphosphate, floats in and binds to the myosin head. This is a critical point to understand. ATP is not used to make the pull. ATP is used to break the grip. The arrival of the ATP molecule changes the shape of the myosin head again. It destabilizes the bond. The magnet is turned off. The myosin head releases the actin. The bridge is broken. The two filaments are separated again. Step 4. Cocking of the myosin head. The myosin is now free, but it is limp. It is in its post-stroke position. It needs to be reset so it can grab a new piece of rope further down the line. The myosin head possesses an enzyme capability. It is an ATPase. It takes that fresh ATP molecule it just caught and breaks it down, hydrolysis. It splits the ATP into ADP and phosphate. Breaking this chemical bond releases a burst of energy. The myosin head absorbs this energy. It uses it to swing back to its original, cocked position. Imagine stretching a rubber band or resetting the mousetrap. The energy is not gone. It is stored inside the structure of the myosin head. It is now tensioned, energized, and reaching out, ready to start the cycle all over again. And so, we are back at the beginning. If calcium is still present, the myosin will instantly grab the next actin pearl, and the cycle will repeat. Attach, pull, release, reset. Attach, pull, release, reset. Now, I want you to multiply this. A single muscle fiber contains billions of myosin heads. If they all pulled and let go at the exact same time, the movement would be jerky. It would be like a tug-of-war team where everyone lets go of the rope simultaneously. The rope would slip back. But nature is smarter than that. The myosin heads work asynchronously. While some are attaching, others are pulling. While some are pulling, others are letting go and resetting. It is like climbing a rope hand over hand. You always have one hand gripping the rope while the other reaches up for a new grip. This ensures that the tension is constant and smooth. The actin filament is pulled inward in a steady, gliding motion. As this happens in thousands of sarcomeres end to end, the Z-discs are drawn closer and closer together. The H-zone in the center disappears. The I-bands shorten. The entire muscle fiber shortens. And remarkably, through all of this, the filaments themselves never change length. They simply slide deeper and deeper into each other's embrace, creating the density and firmness you feel in a contracted muscle. This mechanism also explains the phenomenon of rigor mortis with crystal clarity. We learned that ATP is required for the myosin to let go of the actin. When a creature dies, the body stops producing ATP. The calcium leaks out, allowing the myosin to grab the actin. Step 1. The myosin pivots and pulls. Step 2. But then, nothing. There is no fresh ATP to bind to the head. Step 3 cannot happen. The myosin cannot detach. It remains locked in the contracted position. The cross bridges become permanent, rigid links. The muscle becomes stiff as stone. It is only days later, when the proteins themselves begin to decompose, that the stiffness fades. It is a stark illustration of how much biological effort is required not just to work, but to let go. Softness is an active state of energy renewal. Let us also consider the concept of the length-tension relationship. Because this mechanism relies on the overlap of filaments, the starting length of the muscle matters. Imagine trying to pull a rope if your hands are already bumping into your chest. You have no room to pull. This happens if a muscle is already fully shortened. It cannot generate much more force. Conversely, imagine trying to pull a rope that is just out of reach of your fingertips. You can't grab it. This happens if a muscle is stretched too far. The myosin heads and actin strands barely overlap, and they cannot find each other to form bridges. The muscle is strongest at an optimal resting length, where there is a nice, healthy overlap, enough room to pull, but plenty of heads ready to grab. Your body naturally maintains your muscles near this optimal length, keeping you ready to move with maximum efficiency. Now, let us integrate this into a single visualization to rest your mind. Imagine a great galley ship rowing through a calm sea. The actin filament is the water. The myosin heads are the oars. The calcium is the captain shouting the command to row. The ATP is the food that feeds the rowers. The rowers reach out, attach. They heave back with all their might, power stroke, propelling the ship forward. They lift their oars out of the water, detach. They push their oars forward again, preparing for the next stroke, reset. Thousands of oars, dipping and rising in a chaotic but productive rhythm. The ship, the muscle, glides forward smoothly, powerfully, shortening the distance to its destination. This is happening in you right now. Even as you sleep, the muscles of your diaphragm are gently cycling through this process. The phrenic nerve fires. Calcium is released. Myosin heads in your diaphragm grab the actin. They pull. Your chest cavity expands. Air rushes into your lungs. Then, the nerve stops. The calcium is pumped away. The myosin lets go. The muscle relaxes. The air leaves your lungs. A cycle of life, powered by the sliding of proteins smaller than a wavelength of light. It is a mechanism of profound elegance. It does not use explosions or combustion. It uses shape. It uses geometry. It uses the tiny attractions between molecules to generate the force that builds civilizations. But we must ask one final question. We have seen that this process is hungry. Every single myosin head needs a new molecule of ATP for every single stroke. And there are billions of heads, and they cycle many times a second. The demand for ATP is astronomical. Where does all this energy come from? The muscle only stores a few seconds worth of ATP at any given time. If it runs out, the machine stops. To keep the ship moving, the body must have massive fuel refineries working in the background, constantly churning out new ATP to replace what is used. This leads us to our next lesson. We must leave the mechanics of the fiber and enter the boiler room. We must understand muscle metabolism. In our next session, we will explore how your body turns the food you eat and the air you breathe into the raw energy needed to cock the myosin gun. We will learn about the different fuel tanks, creatine, glucose, and oxygen, and how your body switches between them depending on whether you are sprinting for a bus or sleeping in your bed. For now, let the oars rest. Let the filaments slide apart to their resting length. Let the cross bridges disconnect. Feel the softness return to your limbs as the chemistry quiets down. You are a vessel of incredible design, functioning perfectly even in your stillness. Drift on that thought. Lesson 7. Muscle metabolism and energetics. Welcome back. Take a moment to find your center. Allow the weight of your body to sink into the mattress, and let your breathing settle into a slow, effortless rhythm. You are safe here, in this quiet sanctuary of learning. There is no rush, no pressure to memorize, only a gentle invitation to understand the workings of your own vessel. In our last session, we witnessed the mechanical miracle of the sliding filaments. We saw the billions of tiny myosin heads reaching out, grabbing the actin ropes, and pulling with all their might to shorten the muscle. It was a picture of incredible coordinated effort, but as with all effort in the universe, it comes at a cost. Nothing moves without payment. Tonight, we must look at the bill. We are going to explore muscle metabolism. We are going to step into the boiler room of the body to see how it generates the fuel that keeps those myosin heads moving. It is a story of chemistry, of timing, and of the precious air we breathe. You may recall from our previous lesson that the myosin head needs a specific molecule to let go of the actin and reset itself. That molecule is ATP adenosine triphosphate. ATP is the universal currency of life. It is the only form of energy that your muscle fibers can directly use. You might eat bread, or steak, or sugar, but your muscle fibers cannot use a molecule of steak. They cannot use a molecule of glucose directly. Everything you eat must be broken down, processed, and transformed into this single, golden coin of ATP. Here lies the challenge. ATP is a heavy, unstable molecule. It is like a fully charged battery that leaks if you let it sit too long. Because of this, your muscle cells cannot store very much of it. In fact, the amount of ATP floating around in your muscles right now, the cash on hand, is enough to sustain contraction for only about 4 to 6 seconds. Think about that. If you were to stand up and sprint as hard as you could, you would burn through your entire stored reserve of ATP before you even cross the living room. Yet, we know you can run for much longer than 6 seconds. You can walk for hours. You can work all day. How is this possible? The answer is that your muscles are not just consumers of energy, they are factories that regenerate it. As soon as you start moving, your muscles begin a frantic, sophisticated process of making new ATP to replace what is being used. They have 3 distinct systems for doing this, 3 different engines, if you will. One is for explosive speed, one is for high-intensity surges, and one is for long, steady endurance. Your body seamlessly switches between them, often without you ever noticing. Let us explore these 3 pathways, from the fastest to the slowest. The first engine is called direct phosphorylation. It involves a molecule with a very special job, creatine phosphate. Imagine you are standing at the starting line of a race. The gun goes off. You explode forward. In those first few seconds, your muscles are screaming for energy. The stored ATP is gone instantly. The other engines, the ones that burn sugar or fat, haven't had time to warm up yet. They are too slow. You need energy now. This is where creatine phosphate saves the day. It is a high-energy molecule stored in your muscles. It acts like a reserve battery. It holds a phosphate group that it is desperate to give away. As soon as your ATP is broken down into ADP, adenosine diphosphate, the creatine phosphate swoops in. It hands its phosphate group to the ADP, instantly turning it back into ATP. It recharges the dead battery in a fraction of a second. This reaction is incredibly fast. It requires no oxygen. It is the most immediate source of power you have. However, like a nitrous oxide boost in a race car, it is very short-lived. Your muscles only store enough creatine phosphate for about 15 seconds of maximum effort. This is the energy system of the weightlifter performing a single heavy squat. It is the energy of the 100-meter sprinter. It is the surge you feel when you leap to catch a falling glass. It is powerful, instant, and fleeting. Once the creatine is used up, the body must switch to a different fuel source. This brings us to the second engine, anaerobic glycolysis. We are now past the 15-second mark. The creatine is gone, but you are still running. Your body needs to find a new way to make ATP, and it needs to do it quickly. It turns to the sugar stored in your blood, glucose, or the sugar stored in your muscles, glycogen. The word glycolysis literally means sugar breaking. In this process, the cell takes a molecule of glucose and snaps it in half. This breaking of bonds releases energy, which is used to rebuild ATP. The defining characteristic of this engine is that it is anaerobic. This means without air. It does not require oxygen to work. This is crucial because when you are working hard, say, running up a steep hill or swimming a fast lap, your heart and lungs might not be able to deliver oxygen to your muscles fast enough to keep up with the demand. You are breathless. You are in a deficit. Anaerobic glycolysis allows you to keep moving despite this lack of oxygen. However, there is a catch. This system is fast, but it is inefficient. For every molecule of sugar it burns, it only gets a tiny amount of ATP, about 2 ATP per glucose. It is a dirty engine. It burns fuel rapidly and leaves behind a heavy exhaust. That exhaust is pyruvic acid, which is quickly converted into lactic acid. You have likely felt the presence of this system before. It is that burning sensation in your muscles when you lift a weight for many repetitions, or when you sprint until your legs feel like lead. That burn is the buildup of acidity. For a long time, people blamed lactic acid for muscle soreness days after a workout. But we now know that isn't true. Lactic acid is simply a byproduct of this fast-burning engine. It changes the pH of the muscle, making it acidic. This acidity interferes with the enzymes and the calcium binding we discussed in earlier lessons. It gums up the works. Eventually, the acidity becomes too high, the enzymes stop working, and the muscle physically refuses to contract. This is the failure point in exercise. So, this second system buys you time. It can sustain high-intensity activity for about 30 to 40 seconds, maybe a minute if you are tough. It bridges the gap between the instant burst of creatine and the long-term endurance engine. And that brings us to the third and final engine, aerobic respiration. This is the masterpiece of metabolism. This is the engine that keeps you alive right now as you lie in bed. It is the engine that powered our ancestors as they walked across continents. Aerobic means with air. This process takes place inside the mitochondria, the kidney bean-shaped power plants found inside your muscle fibers. In this system, the muscle takes glucose, or fatty acids, or even amino acids from proteins, and feeds them into the mitochondria. Inside this microscopic furnace, the fuel is mixed with oxygen. Because oxygen is present, the fuel can be burned completely. It is not just snapped in half like in glycolysis, it is disassembled atom by atom. The energy yield is tremendous. Remember that the anaerobic system squeezed only 2 ATP out of a sugar molecule? The aerobic system, using oxygen, can squeeze 32 ATP out of that same sugar molecule. It is 16 times more efficient. And the waste products? They are harmless. Carbon dioxide and water. This system is clean, high yield, and sustainable. It can run for hours. As long as you can breathe fast enough to supply the oxygen, and as long as you have fat or sugar to burn, this engine will keep going. This is the system used by the marathon runner, the cyclist, and the sleeper. The only downside is that it is slow to start. It involves many complex chemical steps, a cycle known as the Krebs cycle, and the electron transport chain. It takes a few minutes for the heart to pump enough oxygenated blood to the tissues to get this factory running at full capacity. This is why, when you start a jog, the first few minutes feel difficult. You are in a transition zone, relying on the inefficient anaerobic systems while the aerobic engine warms up. Once you hit your stride and your breathing settles, you have entered the aerobic zone. You are cruising. Let us pause and visualize these three systems working together in a relay race. Imagine a tennis player. She stands ready at the baseline. Suddenly, She lunges for a serve. That explosive lunge? That is creatine phosphate. Instant power. Now, a long rally begins. She is sprinting back and forth, hitting hard forehands. The point drags on for 45 seconds. Her lungs are burning. Her legs feel heavy. She is now relying on anaerobic glycolysis. The acidity is rising. Finally, the point ends. She walks slowly back to the line. She breathes deeply. Her heart rate is high, but she is recovering. During this low-intensity walking, and for the rest of the match, her baseline energy is provided by aerobic respiration, burning fats and sugars cleanly to recharge her for the next burst. It is a seamless dance. The body does not abruptly switch from one to the other. It blends them, always seeking the most efficient way to meet the demand of the moment. But what happens when the demand is too high for too long? We experience muscle fatigue. Fatigue is a safety mechanism. It is the inability of the muscle to contract effectively despite being told to do so. It is not just about running out of ATP. In fact, total ATP depletion rarely happens. If it did, the muscle would lock up in rigor, as we discussed. Instead, fatigue is usually caused by ionic imbalances. Remember the sodium and potassium pumps? Remember the calcium flood? During intense exercise, these ions get messy. Potassium accumulates in the fluids. Calcium regulation gets sloppy. The intricate electrochemical balance is disturbed, and the muscle simply loses its ability to listen to the nerve. It protects itself by shutting down before it causes damage. And finally, we must address the aftermath. You have finished your exercise. You sit down to rest, but you do not stop breathing heavily immediately. You continue to pant for several minutes, or even hours, after the work is done. Why? This phenomenon is called excess post-exercise oxygen consumption, or EPOC. It is more commonly known as the oxygen debt. You are paying back the loan. During those first few minutes of running, when you were using the anaerobic systems, you borrowed energy. You created lactic acid. You used up your creatine reserves. You drained the oxygen stored in your myoglobin. Now, you must restore the balance. The heavy breathing provides the extra oxygen needed to convert the toxic lactic acid back into useful glucose in the liver, resynthesize the creatine phosphate reserves in the muscle, restock the oxygen on the myoglobin molecules, and simply to support the elevated metabolic rate that comes from a hot body temperature. Your body is tidying up the factory. It is restocking the shelves, sweeping the floor, and cooling down the machinery so that it is ready for the next day. So, as you lie here tonight, consider the quiet, aerobic hum of your own metabolism. You are in the ultimate steady state. Your breathing is slow because your demand is low. Your mitochondria are happily sipping oxygen, burning mostly fatty acids, the slow-burning logs of the metabolic fire, to generate the steady stream of ATP needed to keep your heart beating, your diaphragm moving, and your cells repairing. There is no burn. There is no rush. There is only the efficient, clean energy of life sustaining itself. This understanding of energy leads us to a fascinating realization. If we all have these three systems, why are some people better at sprinting, while others are born to run marathons? The answer lies in the fibers themselves. Not all muscle fibers are built the same. Some are designed to hold more creatine. Others are packed with mitochondria. Some are built for the dash, others for the distance. In our next lesson, we will look at these differences. We will explore contraction types and muscle fiber classification. We will learn why a chicken has dark meat and white meat, and why you are a mixture of both. For now, let the chemistry settle. Let the fires of the mitochondria burn gently in the background, keeping you warm. Let the lactic acid wash away. Let the creatine recharge. Rest in the knowledge that your internal engines are running perfectly, silently, and efficiently, carrying you through the night. Lesson 8: Contraction Types and Muscle Fiber Classification. Welcome back to our fireside. It is good to have you here again. Take a moment to adjust your pillow, to loosen the blanket, and to let the muscles of your face and neck soften completely. You have been a diligent student of your own body, traveling from the gross anatomy of the limbs down to the microscopic sliding of protein filaments. You have watched the sparks of the nerves and stoked the fires of metabolism. Tonight, we are going to look at the nuance of movement. Up until now, we have spoken of muscle contraction as if it were a single, simple event. The muscle shortens and the bone moves. But the reality is far more subtle and sophisticated. Did you know that a muscle can contract without moving at all? Did you know it can contract while getting longer? And did you know that the muscles in your back are made of a fundamentally different material than the muscles in your eyelids? Tonight, we explore contraction types and muscle fiber classification. This is a lesson about diversity. It is about understanding that your body is not built of one generic material, but of a specialized mosaic of tissues, each tuned for a specific kind of music, some for the slow, steady rhythm of endurance, and others for the explosive crescendo of power. Let us begin by redefining what we mean by contraction. In common language, we think contraction means shortening, but in physiology, contraction simply means the generation of tension. It means the myosin heads are grabbing the actin and pulling. What happens next, whether the muscle actually changes length, depends on the relationship between the force you generate and the weight you are trying to lift. We classify these efforts into two main categories: isotonic and isometric. Let's start with isotonic contractions. The word comes from Greek roots. Iso means same, and tonos means tension. In an isotonic contraction, the tension in the muscle remains constant, but the muscle changes length. This is what you usually think of as exercise: moving a load, walking, lifting a cup, swinging a door. But within this category of movement, there are two distinct phases, like the inhalation and exhalation of a breath. The first phase is the concentric contraction. This is the lifting phase. Imagine you are holding a heavy book in your hand and you decide to curl it up toward your shoulder. Your biceps muscle contracts. It generates enough force to overcome the weight of the book. As a result, the muscle shortens. The Z discs we discussed earlier are pulled closer together. The myosin heads win the tug of war. The angle of your elbow closes. This is a concentric contraction. The muscle shortens while doing work. But what goes up must come down. Imagine now that you are slowly lowering that heavy book back to the table. You don't just drop it, you lower it with control. Your biceps is still working. If it relaxed completely, the book would crash down. So, the muscle is still contracting, still generating tension to fight gravity. But the weight of the book is stronger than the pull of the muscle. The muscle is physically lengthening while it is trying to contract. The myosin heads are grabbing and pulling, but the load is dragging them apart. They are acting as a brake. This is called an eccentric contraction. Eccentric contractions are fascinating. They are about control. They allow you to walk downstairs without tumbling. They allow you to set down a glass without breaking it. Interestingly, eccentric contractions generate much more force than concentric ones. They are also the primary cause of that soreness you feel a day or two after a new workout. This is because the act of braking, of holding on while being stretched, causes tiny, harmless micro tears in the muscle fibers. This micro damage is actually the signal for the body to repair and build the muscle stronger. So, the lowering phase is just as important as the lifting phase. Now, let us shift to the second major category: isometric contractions. Iso means same, and metric refers to length. Same length. Imagine you walk up to a solid brick wall. and push against it with all your might. Your muscles are firing. Your metabolism is burning fuel. The myosin heads are grabbing and pulling furiously. You can feel the tension building in your chest and arms, but the wall does not move. Because the load, the wall, is greater than the force you can generate, the muscle does not shorten. It stays the exact same length. The sarcomeres are generating force, but they aren't sliding anywhere. This might sound useless, but isometric contraction is actually the bedrock of your existence. It is the contraction of stillness. Right now, even as you lie there, the muscles of your neck are likely making tiny isometric adjustments to keep your head comfortable. When you stand upright, the muscles of your back and legs are in a constant state of isometric contraction to keep you from collapsing under gravity. They are holding the line. Isometric contractions stabilize your joints. They maintain your posture. They provide the solid foundation from which all other movement occurs. Without these silent, static contractions, we would be like marionettes with our strings cut. So, visualize the difference. Concentric is the lifting, the shortening, the victory over gravity. Eccentric is the lowering, the lengthening, the controlled surrender to gravity. Isometric is the holding, the stillness, the unwavering resistance. Shutterstock. Now that we understand how muscles contract, let us look at what they are made of. If you have ever looked at a Thanksgiving turkey, you know that there is white meat and dark meat. The breast meat is white. The leg meat is dark. This is not a culinary accident. It is a lesson in physiology. Your muscles follow the exact same pattern. Human muscles are a mixture of different fiber types. We classify them based on two things: how fast they twitch, speed, and how they make their ATP, metabolism. Let us meet the characters in this cast. First, we have the slow oxidative fibers. We also call them type I, or slow twitch fibers. These are the dark meat. They are red in color. Why red? Because they are packed with myoglobin, that oxygen storage protein we mentioned earlier, and they are rich with capillaries, the tiny blood vessels that deliver oxygen. These fibers are the endurance specialists. They are small and thin. They are not very powerful. However, they are incredibly efficient. They are packed with mitochondria. They rely on aerobic respiration, the clean, efficient burning of fats and sugars using oxygen. Because of this, they are extremely resistant to fatigue. They can contract for hours and hours without getting tired. These are the fibers that maintain your posture. The muscles along your spine that keep you upright all day long are made mostly of these slow oxidative fibers. They are the marathon runners of your body, slow, steady, and relentless. On the other end of the spectrum, we have the fast glycolytic fibers. We call them type 2B, or fast twitch fibers. These are the white meat. They are pale in color. They have very little myoglobin and very few capillaries. They don't need much oxygen because they don't use it. These fibers are the powerhouses. They are large in diameter. They are packed with glycogen, sugar, fuel. Their myosin heads move incredibly fast. They generate massive amounts of force. When you swing a baseball bat, or jump for a rebound, or lift a heavy box, you are recruiting these fibers. But there is a price. Because they rely on anaerobic glycolysis, the sugar snapping, lactic acid producing engine, they tire very quickly. They are like a drag racer: huge engine, tiny gas tank, lots of exhaust. They burn bright and burn out fast. And finally, nestled between these two extremes, we have the fast oxidative fibers, type 2AA. These are the middle ground. They are pink, they have some myoglobin and capillaries, but not as much as the slow fibers. They can use oxygen, but they can also switch to anaerobic pathways. They are faster than the slow fibers, but more enduring than the fast fibers. They are the walking and sprinting fibers. versatile, and adaptable. Now, pause and consider the distribution of these fibers in your own body. Most of your muscles contain a mixture of all three types. It is like a team composed of heavy lifters, sprinters, and distance runners. However, the ratio changes depending on what the muscle does. Your eye muscles, which need to dart back and forth instantly, are mostly fast twitch. Your soleus muscle in your calf, which holds you up while you stand, is mostly slow twitch. Your biceps, which does a bit of everything, is a fairly even mix. But what determines your specific mix? This brings us to the question of genetics versus training. The truth is, much of your fiber composition is determined before you are even born. It is written in your DNA. Some people are born with a high percentage of slow oxidative fibers. These people are natural endurance athletes. They can run forever without tiring, but they might struggle to put on massive muscle bulk or sprint with explosive speed. They are built for the long haul. Others are born with a dominance of fast glycolytic fibers. These are the natural sprinters and powerlifters. They are explosive and strong, capable of building large, powerful muscles, but they might find a long jog exhausting. This is the hand nature deals you. However, nurture your habits can influence the game. This is the concept of plasticity. Your muscles can change. While you cannot turn a slow fiber completely into a fast fiber, or vice versa, you can shift their characteristics. If a person with average genetics trains for a marathon, their fast fibers will start to look and act more like slow fibers. They will grow more mitochondria. They will develop more capillaries. They will become more efficient. Conversely, if you spend your time lifting heavy weights, your muscle fibers will adapt by becoming larger and storing more glycogen, shifting toward the powerful, glycolytic end of the spectrum. Your body is constantly listening to what you ask of it and remodeling itself to meet the demand. Let's take a moment to visualize this symphony of recruitment. Imagine you are about to pick up an object, but you don't know how heavy it is. You reach out. Your brain first recruits the small, slow, oxidative fibers. They are the cheapest to run. They engage gently. If the object doesn't move, your brain realizes it needs more power. It calls in the fast oxidative fibers. The tension rises. If the object is still too heavy, perhaps it is a box of books, your brain finally calls in the big guns, the fast glycolytic fibers. The explosive giants wake up. They generate the maximum force needed to lift the load. This is called the size principle of recruitment. Your body always uses the most energy-efficient fibers first, and only calls on the expensive, easily fatigued fibers when absolutely necessary. It is a strategy of conservation. So, as you lie here, resting in the quiet dark, think about the fibers that make up your form. Think of the slow fibers in your back, quietly humming along, maintaining your alignment on the bed with barely a whisper of effort. Think of the fast fibers in your legs and arms, currently dormant, their glycogen tanks full, waiting for the moment you need to move quickly. They are all there, a diverse community of cells, each with a specialized role, waiting for your command. It is a comforting thought, isn't it? To know that you are built for both the sprint and the marathon. You are built for the heavy burden and the light touch. You are built for the motion of the concentric and the control of the eccentric. You are a vessel of infinite potential adaptability. We have now explored the voluntary world of skeletal muscle in great depth. We know its structure, its chemistry, and its types. But we must remember that there are muscles in your body that do not wait for your command. They do not care about your workout routine. They are the silent guardians that work while you sleep. In our next lesson, we will leave the skeletal system behind and visit the autonomic world. We will journey to the heart and the hollow organs to meet the cardiac and smooth muscles. We will learn why the heart never tires and how the stomach knows how to digest without you ever teaching it. For now, let your own muscles soften. Release any isometric tension you might be holding in your jaw or shoulders. Let gravity take over. Drift into the stillness, where no force is required and all fibers can rest. Lesson 9. Cardiac and smooth muscle tissue. Welcome back. Listen to that rhythm in your mind for a moment. Thump, 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 thump. It is the oldest sound in your world. It began before you were born, and it continues, steady and true, through every moment of your sleep and every second of your waking life. In our previous sessions, we spent a great deal of time with the skeletal muscles, the muscles of the will. We explored the biceps, the quadriceps, the muscles you command to lift, to run, and to build. These are the muscles of action. They wait for your order, and they obey your intent. But tonight, we are going to leave the realm of the voluntary. We are going to journey into the silent, automatic world of the body's internal guardians. We are going to visit the muscles that work without you ever asking them to. They are the caretakers of your life, operating on a wisdom that runs deeper than conscious thought. We are speaking, of course, of cardiac muscle and smooth muscle. These two tissues control the flow of your blood, the digestion of your food, the pressure in your arteries, and the rhythm of your existence. They are the autopilot of the human vessel. Let us begin our exploration with the tireless engine in the center of your chest. Part I. Cardiac muscle. Cardiac muscle is a rare and precious material. It is found in only one place in the entire human body, the wall of the heart. In anatomy, we call this layer the myocardium. If you were to hold a heart in your hands, it would feel firm, muscular, and incredibly strong. It has to be. The heart is a pump that must contract roughly 100,000 times a day, every day, for decades, without ever stopping to rest. If a skeletal muscle in your leg were asked to work that hard, it would fatigue and fail within minutes. The heart cannot fail. So, its structure must be fundamentally different. Let us look at it through our imaginary microscope. At first glance, cardiac muscle looks somewhat like the skeletal muscle we have been studying. It is striated. It has those same alternating light and dark bands of actin and myosin filaments. This tells us that it uses the same sliding filament mechanism to generate force. It pulls, it shortens, it creates pressure, but the similarities stop there. Skeletal muscle fibers are long, separate, cylindrical tubes, like individual cables running side by side. Cardiac muscle cells are short, fat, and interconnected. They are branched. Imagine the roots of a tree or the Y shape of a slingshot. One cell splits and connects to two others, which split and connect to two others. This creates a complex, three-dimensional web of tissue, rather than a bundle of straight lines. The most critical feature of cardiac muscle, the secret to its success, lies at the points where these cells meet. If you look closely at the junction between two heart cells, you see a dark, zigzagging line. This is called the intercalated disc. I want you to visualize this disc. It is not just a wall, it is a specialized docking station with two vital components. First, it contains desmosomes. Think of these as molecular rivets or spot welds. They physically lock the two cells together because the heart is constantly filling and squeezing. There is a tremendous amount of mechanical stress. Without these rivets, the cells would simply rip apart under the pressure. The desmosomes ensure the heart remains a solid, unbreaking container. Second, and perhaps more magically, the intercalated disc contains gap junctions. These are microscopic tunnels that punch through the walls of the neighboring cells. They allow the fluid from one cell to flow directly into the next. Why is this important? Remember that muscle contraction is triggered by electricity, by the flow of ions like sodium and calcium. In skeletal muscle, every cell needs its own nerve connection to get the spark. But in the heart, the gap junctions allow the spark to travel from cell to cell on its own. When one cell gets excited and contracts, it instantly passes that electrical fire through the tunnel to its neighbor, who passes it to the next. The result is that the wave of contraction sweeps across the heart in a perfectly coordinated ripple. The heart contracts as a single unit, or what physiologists call a functional syncytium. It does not twitch in parts. It beats as a whole. Now, let us discuss the brain of the heart. Skeletal muscle is paralyzed without a nerve to tell it what to do. If you cut the nerve to your leg muscles will never move again. Cardiac muscle is different. It possesses the property of automaticity or autorhythmicity. This means the heart can beat entirely on its own. It does not need the brain. In fact, if you were to remove a heart from the body and keep it supplied with oxygen and nutrients, it would continue to beat. This is because of a special group of cells called the pacemaker cells. These cells are not stable. Their electrical charge naturally drifts upward until it hits a threshold and fires. Then it resets and drifts up again. This drift creates the rhythm. It is the internal metronome of life. Your brain can speed up the heart when you are scared or slow it down when you are sleeping, but it does not start the heart. The heart owns its own rhythm. Finally, we must look at the fuel source. The heart is an aerobic machine. It is packed with mitochondria far more than skeletal muscle. These power plants take up nearly 25% of the volume of a heart cell. This means the heart is incredibly resistant to fatigue. It is built to burn fuel cleanly and efficiently using oxygen. However, this is also its great vulnerability. Because it relies so heavily on oxygen, it cannot function well without it. Skeletal muscle can work anaerobically without oxygen for a while when you sprint. The heart cannot. If the blood supply is cut off, as happens in a heart attack, the muscle cells begin to die very quickly. They starve for oxygen. It is the price paid for endless endurance. Part 2. Smooth muscle. Now, let us shift our gaze. We leave the chest and travel to the rest of the body's interior. We go to the stomach, the intestines, the bladder, the uterus, and the lining of the blood vessels. Here, we find smooth muscle. This tissue gets its name because, under the microscope, it has no stripes. It is non-striated. It appears smooth, uniform, and silky. Why no stripes? Because the actin and myosin filaments inside are not arranged in those rigid, military-style sarcomeres we saw earlier. There are no Z-discs. There is no regular pattern. Instead, the filaments are arranged diagonally, crisscrossing the cell like a fishnet stocking. They are anchored to the cell membrane by structures called dense bodies. The cells themselves are small and spindle-shaped, thick in the middle and tapered at the ends, like a grain of wheat or a kayak. They have a single nucleus in the center. The movement of smooth muscle is fundamentally different from the others. When skeletal muscle contracts, it shortens in a straight line, like a telescope collapsing. When smooth muscle contracts, it twists. Because of that fishnet arrangement of fibers, when the filaments pull, the whole cell corkscrews. It wrings itself out like a wet towel being twisted dry. This twisting action is perfect for its job. Smooth muscle usually wraps around tubes or sacs. When it twists and squeezes, it constricts the tube. It pushes the contents forward. This leads us to the two main superpowers of smooth muscle, peristalsis and endurance. Peristalsis is the rhythmic wave of contraction that moves food through your digestive tract. Imagine a tube of toothpaste. If you squeeze at the bottom and slowly slide your thumb up, the toothpaste moves out. In the intestine, Smooth muscle is arranged in two layers. One layer runs in a circle around the tube, circular layer. The other runs lengthwise along the tube, longitudinal layer. By coordinating these two layers, squeezing the ring behind the food, and shortening the tube in front of it, the body propels the food forward. It is a dance of mixing and pushing that happens automatically, especially while you sleep. But smooth muscle has another incredible ability: the stress relaxation response. Think about your bladder or your stomach. These are organs that need to fill up. If skeletal muscle were stretched, it would bounce back immediately. But when smooth muscle is stretched, say, as your stomach fills with a large meal, it briefly resists, but then it relaxes and adapts to the new size. It allows the organ to expand without increasing the pressure inside. It makes room for storage. It is patient. And then there is its efficiency. Smooth muscle is the tortoise of the body. It is slow to contract and slow to relax. A skeletal muscle might twitch in a fraction of a second. Smooth muscle might take several seconds to complete a single wave. But it can hold a contraction for a very, very long time, without getting tired and without using much energy. This is called the latch mechanism. Think of the sphincter muscle that keeps your bladder closed. It has to stay squeezed shut for hours at a time. If it burned energy like skeletal muscle, you would be exhausted just from holding your water. But smooth muscle can latch its actin and myosin together, locking them in the contracted position like a parking brake. Once latched, it maintains the tension with almost zero energy cost. It can hold the line indefinitely. Part 3. Control and regulation. So, who is driving these muscles if not you? We said they are involuntary. You cannot sit there and decide to make your stomach churn or your arteries dilate. These muscles are controlled by the autonomic nervous system. This system has two branches, often acting like a gas pedal and a brake. The sympathetic nervous system is the fight or flight response. When you are in danger, it sends signals to speed up the heart, cardiac muscle, and constrict the blood vessels, smooth muscle, to raise blood pressure. At the same time, it tells the smooth muscle in your digestion to stop working, because you don't need to digest lunch while running from a tiger. The parasympathetic nervous system is the rest and digest response. It is the system active in you right now as you relax. It sends signals to slow the heart rate down, conserving energy. It stimulates the smooth muscle of the stomach and intestines to get to work processing nutrients. It opens the blood flow to the skin and digestive tract. But nerves are not the only controllers. Smooth muscle is remarkably sensitive to chemistry. Hormones play a huge role. Adrenaline, released by your adrenal glands, can make your heart race and your airways open up. Gastrin, a hormone in the stomach, tells the smooth muscle there to churn. Even local conditions change the muscle. If a tissue in your body is low on oxygen, the smooth muscle in the nearby arteries will relax, widening the pipe to let more blood flow in. It is a smart tissue. It tastes the environment and adjusts accordingly. Part 4. Synthesis. Let us take a moment to stand back and appreciate the harmony of these three systems working together. Visualize the skeletal muscle, the movers, the soldiers. They are big, fast, and powerful. They act on the world outside. They are expensive to run, and they need your constant guidance and command. Visualize the cardiac muscle, the timekeeper, the drummer. It is a unified web of electrical connection. It is relentless, beating once per second for a lifetime, never stopping, fueling itself with a constant stream of oxygen. It connects the timeline of your birth to the timeline of your death. Visualize the smooth muscle, the regulator, the adjuster. It lines the hollow spaces. It is slow, efficient, and infinitely adaptable. It manages the flow of life, the air, the blood, the food. the waist. It holds the doors shut and pushes the river forward. As you lie in bed tonight, realize that you are trusting your life to these involuntary muscles. And it is a trust well placed. You do not need to stay awake to remind your heart to beat. You do not need to concentrate to keep your blood pressure stable. You do not need to think about moving your dinner through your intestines. Your body has automated the essentials. It has freed your mind to dream, to think, and to rest by assigning the critical tasks of survival to these tireless, intelligent tissues. In the silence of your room, you might feel the pulse in your neck. That is the cardiac muscle pushing. You might hear a faint gurgle in your abdomen. That is the smooth muscle pushing. They are the background music of your existence. We have now met all the players. We know the voluntary and the involuntary. We know the movers and the shakers. But in the real world, muscles rarely work in isolation. A single step, a single smile, or a single breath involves a complex choreography of many muscles working together, some pulling, some relaxing, some stabilizing. To finish our course, we must look at the team dynamics. In our final lesson, Lesson 10, we will explore the interactions of skeletal muscles. We will learn about prime movers, antagonists, and synergists. We will see how the body solves the physics of leverage to turn a small contraction into a grand movement. For now, surrender control. Let the skeletal muscles go limp. Trust the cardiac muscle to keep the beat. Trust the smooth muscle to manage the flow. You are a passenger in a vehicle that knows exactly how to drive itself. Rest in that safety. Lesson 10. Interactions of skeletal muscles. Welcome. Welcome to our final session. Take a deep breath in, filling your lungs completely, and then let it go with a slow, deliberate sigh. Feel the mattress supporting you. Feel the blanket settling around you. You have traveled a long way with me. We have journeyed through the hidden landscapes of your own body, from the microscopic sliding of protein filaments to the silent pumping of the heart. We have taken the machine apart, piece by piece, to understand how it works. Tonight, we put it back together. In this final lesson, we will look at the body not as a collection of separate parts, but as a unified, moving whole. We will explore the interactions of skeletal muscles. We will discover that muscles rarely work alone. In fact, almost every movement you make, whether it is a smile, a step, or a wave of the hand, is a complex symphony performed by a team of muscles working in perfect harmony. The central truth of the muscular system is this: muscles can only pull. They can never push. Think about that for a moment. A muscle fiber is a rope. You can pull on a rope to bring an object closer, but you cannot push a rope. When a muscle contracts, it shortens and pulls a bone. But how does the bone get back to where it started? The muscle cannot push it back. To solve this problem, nature has organized muscles into teams. For every action, there must be a muscle to reverse that action. This simple rule dictates the layout of your entire body. It creates a system of balance, of opposition, and of cooperation. To understand this, let us visualize the movement of your arm. Imagine you are bending your elbow to bring a cup of tea to your lips. In this specific movement, the biceps brachii is the leader. It is the muscle primarily responsible for the action. We call this muscle the prime mover, or the agonist. The word agonist comes from the Greek word for contest or struggle. It is the main contender. It is the one contracting, shortening, and doing the heavy lifting. But look at the back of your arm. There lies the triceps brachii. If the triceps were to contract at the same time as the biceps, your arm would be locked in a rigid stalemate. Both sides would be pulling, and nothing would move. So, for the cup to rise, the triceps must yield. It must relax and stretch to allow the movement to happen. We call this muscle the antagonist. The antagonist opposes the prime mover. But do not think of it as an enemy. Think of it as a partner. It provides a braking force. It controls the movement, ensuring that your arm doesn't snap up too quickly or hyperextend. It keeps the motion smooth and safe. This relationship, the agonist contracting while the antagonist relaxes, is controlled by the nervous system. It is called reciprocal inhibition. It is a neurological wiring that ensures you don't trip over your own feet. When the nerve sends a go signal to the agonist, it simultaneously sends a stop signal to the antagonist. It is a dance of give and take, but the team is larger than just two. Let us look closer at that arm movement. If the biceps were the only muscle working, the movement might be wobbly, or perhaps the force wouldn't be quite right. Enter the synergists. Syn means together, and erg means work. These are the helpers. Synergists are smaller muscles that assist the prime mover. They might add a little extra force to the pull, or they might help guide the movement to ensure it follows a straight path. They are like the supporting violins in an orchestra, adding depth and stability to the melody played by the soloist. And finally, we have the unsung heroes of movement, the fixators. Imagine you are trying to open a heavy door. You plant your feet firmly on the ground to get leverage. If your feet slipped, you couldn't pull the door. Your muscles need a solid base too. When your biceps contracts, it pulls on the radius bone in your forearm, but it also pulls on the scapula, the shoulder blade, where it originates. If the shoulder blade were loose, the biceps would just pull the shoulder forward instead of lifting the arm. So, the fixator muscles like the rhomboids and the trapezius in your back contract isometrically. They lock the shoulder blade in place. They bolt it to the spine. They create an immovable anchor so that all the force of the biceps can be directed into lifting the cup. Fixators stabilize the origin so the prime mover can move the insertion. They are the foundation of strength. So, for a simple sip of tea, the fixators lock the shoulder. The agonist, biceps, lifts the forearm. The synergist, brachialis, helps the lift. The antagonist, triceps, relaxes and guides the speed. 4 distinct roles, played by different muscles, all happening in a split second without you ever thinking about it. Now, let us shift our perspective from biology to physics. The skeleton and muscles interact to form lever systems. Your body is essentially a machine made of levers. You are living proof of the principles of mechanics. A lever system has 4 parts. The lever, a rigid bar. In your body, this is the bone. The fulcrum, a fixed point around which the lever moves. In your body, this is the joint, the effort, the force applied to move the lever. This is the muscle contraction, the load, the resistance being moved. This is the weight of your limb, or the object you are lifting. Depending on how you arrange these parts, the joint, the muscle, and the weight, you get different results. You can trade power for speed, or speed for power. Let's look at the 3 classes of levers in your body. First-class levers. Imagine a seesaw or a pair of scissors. The fulcrum is in the middle, with the effort on one side and the load on the other. We have only a few of these in the body. The classic example is your head sitting on your neck. The joint between the skull and the spine is the fulcrum. The weight of your face is the load, trying to tip your head forward. The muscles in the back of your neck provide the effort, pulling down to keep your head upright. It is a system designed for balance. Second-class levers. Imagine a wheelbarrow. The wheel, fulcrum, is at the front. The heavy load is in the middle. You lift the handles, effort, at the back. This is a lever of immense strength. You can lift a huge load with a small effort. In the body, this is found when you stand on your tiptoes. The ball of your foot is the fulcrum. The weight of your entire body is the load in the middle. The calf muscles pulling up on the heel are the effort. This design gives your calves the mechanical advantage to lift your whole body weight easily, but the range of motion is small. You can't lift your heels very high. Third-class levers. This is the most common lever in the human body. Imagine a pair of tweezers or a fishing rod. The fulcrum is at one end. The load is at the far other end. The effort is applied in the middle, between them. Let's go back to our biceps. The elbow joint is the fulcrum. The load, the hand, is far away at the end of the lever. The biceps attaches to the forearm just a tiny bit past the elbow. This means the muscle is pulling very close to the joint, while the weight is far away. Mechanically, this seems like a bad idea. It is a mechanical disadvantage. Your biceps has to work much harder to lift the weight than if it were attached near the wrist. So why would nature design us this way? Why choose the difficult path? The answer is speed and range of motion. Because the muscle attaches so close to the joint, a tiny movement of the muscle translates into a huge movement of the hand. If your biceps contracts just 1 centimeter, your hand might swing through an arc of 10 centimeters. This allows you to move your limbs fast. It allows you to throw a ball, to run, to strike. Nature sacrificed raw power to give you speed and agility. It is a brilliant trade-off. Finally, we must consider the shape of the muscles themselves. Just as the arrangement of the lever matters, the arrangement of the fascicles, the bundles of fibers, matters. Some muscles are parallel. The fibers run straight from end to end like a strap. The sartorius in your thigh is a great example. These muscles can shorten a lot. They have great range of motion, but they are not very strong. Other muscles are pennate. This means feather-shaped. The fibers run at an angle to the tendon, just like the barbs of a feather attached to the central quill. This design allows the body to pack thousands more muscle fibers into the same space. More fibers mean more power. The quadriceps and the deltoids are pennate muscles. They cannot shorten as much as the parallel muscles, but they are incredibly strong. Again, we see the trade-off. Parallel for reach, pennate for power. Conclusion of the course. And so, we have reached the end of our curriculum. Let us take a moment to look back at the path we have walked together. It is a journey from the infinitely small to the beautifully large. We began with the tissues, learning the difference between the voluntary skeletal muscle, the rhythmic heart, and the enduring smooth muscle. We explored the gross anatomy, peeling back the epimysium to find the fascicles within. We shrank down to the microscopic level, standing inside the sarcomere to witness the geometry of the Z-discs. We watched the spark of the neuromuscular junction, where the chemical acetylcholine bridged the gap between thought and action. We followed that spark through the tubules to the calcium flood, unlocking the doors of the protein guards. We marveled at the sliding filament mechanism, the hand-over-hand climbing of billions of myosin heads. We visited the boiler room of metabolism, understanding how creatine, sugar, and oxygen fuel the fire. We categorized the fiber types, the sprinters, and the marathoners living side by side in your limbs. We honored the silent, automatic work of the cardiac and smooth systems. And tonight, we saw how they all join hands, agonist, antagonist, synergist, and lever, to create the miracle of movement. I hope that through these lessons, you have gained a new perspective on the vessel you inhabit. Your body is not a static object. It is a dynamic event. It is a ceaseless flow of chemistry and physics, governed by laws of balance and economy. It is a structure built for motion, yet capable of profound stillness. As you lie here now, all of these systems are quieting down. The levers are resting. The gravity is doing the work for you. The calcium pumps are slowing their pace. The mitochondria are humming a lullaby of low-energy maintenance. The symphony has finished its crescendo and now plays a soft, slow adagio for the night. You can trust this machine. It has evolved over millions of years to support you, to move you, and to restore you. You do not need to manage it. You only need to respect it, feed it, and let it rest. Close your eyes, if they aren't already closed. Feel the heaviness of your limbs, those levers of bone and muscle sinking deeper into the bed. Feel the gentle rise and fall of your chest, the work of the diaphragm and intercostals bringing the oxygen that is life. Feel the steady beat of the heart, the tireless drummer keeping watch. The lesson is over. The knowledge is yours. The rest is now. Sleep well and drift into the quiet wisdom of your own design.